Hello students. So today's topic is blood and its components. Let's go into the topic. So what is blood? Blood is a connective tissue in fluid form. It is the fluid of our life. Okay, what is connective tissue? Connective tissues are tissues which connect one part from another. So blood is the connective tissue in fluid form. In human body, the volume of blood is about 5 to 6 liters approximately. In men, it varies from 5 to 5.5 liters, and in women it varies from 4.5 to 5 liters. So the color of the blood is red, but in case of oxygenated blood, is scarlet red color, whereas deoxygenated blood is bluish red or blackish red color. In general terms, blood is red color, whereas oxygenated blood is scarlet red and deoxygenated blood is bluish or blackish red color. So what is the pH of the blood? The blood pH is about 7.4, which is alkaline. So the normal neutral pH is 7, whereas blood pH is slightly alkaline, which is 7.4. And the viscosity of blood is about 5 times more than that of water. The viscosity changes in some diseases like anemia and in dehydration. In dehydration, the viscosity may be more, whereas in anemia, the viscosity will be less. So going to the next slide. So coming to the functions of blood. So I have written a mnemonic for the functions of the blood, DROPT. So let's decode the mnemonic. D for detoxification, R for regulatory functions, O for osmotic balance, P for protective functions, and finally T for thermoregulatory and transport functions. So coming to detoxification, blood is the main medium through which the toxins from various sides of the body gets excreted via kidneys. Next, regulatory function. So through blood, so many hormones and electrolytes will be circulating around different systems of the body only through blood. So these hormones, they regulate various functions inside our body. Coming to osmotic balance, blood has a plasma protein called as albumin, which helps in osmotic balance. So we will discuss the osmotic balance in plasma protein section. Next, protective function. So immune cells, blood contains immune cells which helps in defense mechanisms. Next, thermoregulatory or transport function. This is the important function of blood. So when it comes to transport, it transports oxygen and carbon dioxide. So this is a rough sketch of the lungs. These are the cells. From lungs, the blood transports oxygen to the cells, whereas from the cells, it transports carbon dioxide back to the lungs. So this is the main transport mechanism. Next we will discuss about the composition of blood. Blood contains blood cells, which are also known as formed elements, and plasma. So when blood is centrifuged, it gets divided into 3 different portions. The upper portion is called as plasma, the lower portion is called as, which contains red blood cells, whereas the middle one, which is the white buffy coat, contains WBC. So these two come under formed elements, that is blood cells, whereas this one is called as plasma. Next we will discuss about plasma. Plasma, it is a straw yellow color or straw color fluid which contributes about 55% of the blood volume. So plasma contains 91% water and 9% solids. Among the 9% solids, we have 7% plasma proteins. So plasma proteins are the important components of blood, and 2% inorganic components. These inorganic components include HEN, hormones, electrolytes and nutrients. Hormones which are secreted from the endocrine glands to reach various parts of the body. Electrolytes which include sodium, potassium, bicarbonate and calcium. Nutrients, so the glucose, lipids and proteins absorbed from the intestine are carried to various parts of the body by blood. Next we are coming to the plasma proteins. So there are three important plasma proteins in plasma which are albumin, globulin and fibrinogen. We will discuss about the function of each plasma protein. The function of albumin is to maintain osmotic balance. What is osmotic balance? So coming to this diagram, there are two different types of pressure, osmotic pressure and hydrostatic pressure. In osmotic pressure, the fluids from the cells are drawn into the blood, whereas in hydrostatic pressure, the fluid is drawn outside the blood. So these two pressures must be maintained in a balanced state, and this is maintained by albumin. So this might be an MCQ question. Which of the plasma proteins are responsible for osmotic balance? Albumin. Next, globulins. So immunoglobulins, they have two different functions. One is defense mechanism and transport. So they are responsible for defense against microbes, and also they are also used as transport proteins to transfer nutrients from one part to another part. Finally, we are coming to fibrinogens. Fibrinogens help in clotting mechanisms. First we will discuss about the clotting mechanism. This is a blood vessel. So if there is an injury site, the platelets, there are two different kinds of clotting mechanisms, primary and secondary. In primary clotting mechanism, whenever an injury occurs in the blood vessel, two different types of clotting mechanism occurs, primary and secondary. In primary, the platelets get accumulated in the site of injury, whereas in secondary, along with the platelets, the fibrinogen will also get accumulated. Thus, fibrinogen helps in clotting mechanisms. Coming to the next slide. Next we are coming to formed elements. There are three different types of cells in blood. Red blood cells or erythrocytes, platelets or thrombocytes, white blood cells or leukocytes. We will discuss each of these cells. The blood cells have a common origin. So they are originated from bone marrow. The bone marrow contains precursor cells called hematopoietic stem cells. From the hematopoietic stem cells, two different types of progenitor cells are formed, the myeloid and the lymphoid cells. From the lymphoid progenitor, we have the lymphocytes, whereas from the myeloid cells, we have the other WBCs, which includes megakaryocytes, from which we have the platelets, eosinophils, basophils, erythrocytes, monocytes and neutrophils. Now we will discuss about this. Coming to erythrocytes. So erythrocytes, erythro means red. So that's why the RBCs are otherwise called as erythrocytes. The main structure of the RBCs includes, RBCs are biconcave structures. So they will be having two concave sides on each side. Okay. So RBCs contain no nucleus. They are anucleate cells. You have to remember this point because this is an important MCQ. So they will give list of cells like RBCs, WBCs, platelets and all of the above. Then they will ask which of these cells is anucleate. So the answer will be red blood cells. Next, origin, as we saw, all of the WBCs and RBCs, they will arise from red bone marrow, and the process of production of RBCs is called as erythropoiesis. Erythropoiesis. Erythro means RBC, poiesis means production. Next, lifespan. So this is the life cycle of RBC. RBC is produced from bone marrow. They circulate inside the blood for about 120 days. After the circulation, they reach the spleen or liver and they get destructed. So this is the lifespan of RBC. Coming to the count of RBCs, there are about approximately 25 trillion RBCs in our blood, and the normal range would be 5 to 6 million cells per cubic millimeter of blood. So it varies with men and women. In men it ranges from 5 to 6, whereas in females it will be ranging from 4.5 to 5. So this is similar to that of blood volume. Blood volume also the range will be same. Next function, obviously RBCs, they help in exchange of gases, oxygen and carbon dioxide. So they carry oxygen from the lungs to the tissues and carbon dioxide from the tissues to the lungs. And another function of RBCs is to determine the blood type. Coming to this, so the RBCs, they have surface antigens. Depending upon these antigens, the blood group is decided. So if they have A antigens, the blood group would be A positive. If they have B antigens, the blood group would be B positive. If they have both the antigens, the blood group will be AB positive. And if they have no antigens, the blood group will be O positive. So they help in gaseous exchange and to determine blood group. Next slide. Next, coming to WBC cells. Similarly, WBC cells are also originated from bone marrow, from the same hematopoietic stem cells structure. So we saw RBCs are anucleate, whereas WBCs are nucleated cells. They will be having nucleus and the size is less than that of RBC. So RBC has no nucleus, WBC has nucleus, RBCs are larger than that of WBCs and the count will also be larger than that of WBCs. So this is the basic difference between RBCs and WBCs. The count, coming to the count, we saw that RBCs are in millions. Here WBCs will be in thousands. So the normal count in human body is 4,500 to 11,000 cells per microliter. WBCs are classified into two different types, granulocytes and agranulocytes. Granulocytes contain granules in their cytoplasm, whereas agranulocytes, they don't have any granules. That's why it is agranulocytes. The granulocytes are further divided into basophils, eosinophils and neutrophils, and agranulocytes are further divided into monocytes and lymphocytes. Now let's discuss each of these cells. Coming to neutrophils, so let's discuss about granulocytes. First, neutrophils. Neutrophils are also called as polymorphonuclear leukocytes. So here we can see in the diagram, the nucleus of the neutrophil, it is multi-lobe. It contains more than two lobes in the nucleus. Hence it is called as polymorpho. So the nucleus has more than two lobes. That's why it is called as polymorphonuclear leukocyte. And neutrophils are the most abundant cells in RBC. So the percentage of neutrophils in the blood is 60 to 65%. They will be in large amount when compared to other WBCs. Function of neutrophils. Neutrophils help in acute inflammation. Whenever there is an injury or an infection, they will be the first responders to go to the site and cause inflammation. The nucleus, coming to basophils, basophils are about 0.5 to 1, they contribute about 0.5 to 1% in the blood, and the main point you have to remember about basophils is they have violet color granules which we can see in the microscope, and they produce histamine. Histamine is an important MCQ. So this has been asked in previous questions. Basophils produce histamine. The function of histamine is vasodilation. What is vasodilation? So this is a blood vessel. Whenever there is a infection or injury, the basophils inside the cells, they will release histamine. This histamine will cause dilation of the blood vessel. So the blood vessel gets dilated, which helps the other cells to move out of the blood and reach the injury or infection site. So which cell produces histamine? Basophils produce histamine. Next we are coming to eosinophils. The difference between basophils and eosinophils is basophils have violet color granules, whereas eosinophils have pink color granules. Both of them help in allergic reactions, but eosinophils are more oriented with parasitic infections. So if they ask the question which cell is involved in parasitic infections, we should choose eosinophils. Next we are coming to agranulocytes. There are two different types of cells, monocytes and lymphocytes. So monocytes are the largest WBC. So which is the largest WBC? Monocytes are the largest WBC, and they contribute about 2% to 10% in the blood. The difference between monocytes and lymphocytes is Monocytes have a kidney shaped nucleus, whereas lymphocytes will have an eccentric nucleus. Eccentric means the nucleus will be over the border of the cell, whereas the monocytes will have kidney shaped nucleus. This is the major difference between them. And the mechanism by which the monocytes help in immune response is phagocytosis. So, in the diagram we can see the monocytes. This is a microbe entering the body. The phagocytes, the monocytes will get converted into macrophages. So, the monocytes will get converted into macrophages, which in turn swallows the microbes and destroys it. So, this is the mechanism. Phagocytosis is uptake of the microbes and particles followed by digestion and destruction. Next, lymphocytes. Lymphocytes contribute about 20 to 25% of the blood volume, and as you already saw, the difference between monocytes and lymphocytes is nucleus will be eccentric. So, lymphocytes will be detailedly discussed in lymphatic system because majority of the lymphocytes are present in lymph. Lymph is another body fluid like blood. So, it will be discussed detailed in lymphatic system. Okay, this is a summary of all the WBCs. Neutrophils, multilobed, polymorphonuclear leukocytes, and these are the first responders. Eosinophils, bilobed and has pinkish granules, and they are more accumulated in parasitic infections. Basophils, which are bi or trilobed, have violet color granules, and their response will be allergic and antigen response. And lymphocytes, two different types of lymphocytes, T and B cells, and they will be helping in cell mediated and humoral immunity. So, it will be discussed in lymphatic system. Monocytes, which has kidney shaped nucleus, and the main function is phagocytosis. So, these are the diagrams: multilobed, pinkish granules, violet granules, eccentric nucleus and kidney shaped nucleus. Next, finally, we are going to discuss about platelets. Platelets are also called as thrombocytes. Thrombus means clot. So, platelets help in formation of clot. That is why they are otherwise known as thrombocytes. How thrombocytes are formed? As we already saw, we have the hematopoietic stem cells from bone marrow. From the hematopoietic stem cells, we have myeloid precursors, and from the myeloid precursors, we have megakaryocytes. This megakaryocyte is a large cell. So, the large cell will get divided into multiple fragments, and these fragments are called as platelets. So, platelets are fragments of cytoplasm of megakaryocytes. Like RBCs, platelets are also anucleate cells. Next structure. So, we have two different types of platelets here, the inactivate form, activate form. So, inactivate form will be biconvex, that is nearly lens shaped, whereas the activated form contains multiple projections. So, this is the main difference between activated and inactivated form of platelets, and as we already saw, the main function of platelets is clotting mechanism, which is also known as hemostasis. Hemo means blood, stasis means to stop bleeding. So, they help in clotting mechanism. We saw already about primary and secondary clotting mechanism, which is given in the diagram. So, here we can see there is an injury, the blood is leaking. So, first in the primary clotting mechanism, the platelet reaches the injury site and it forms the platelet plug and followed by fibrinogen, which forms the meshwork. So, this is primary and this is secondary clotting mechanism. To summarize, in the whole session we saw about blood and volume of blood components, which is divided into two different types, plasma and blood cells, and the various types of blood cells we discussed. So, the important points to remember is numerical values. Next, we have to remember functions of each cells. So, coming to values, the one point you have to notice, RBC is, the count of RBC is represented in millions, platelets in thousands, so in lakhs, and finally WBC in thousands. So you should not forget this, and we discussed about various functions of the cells. And next you should remember about the important plasma proteins and their functions, among which albumin is important, which helps in osmotic balance. Okay, that's it for today. So thank you for watching this video.